Yes, please. What are you looking for? With a B?
The things that she
Yeah, it's.
fair, I think, sometimes. Yeah.
I thought I saw it.
My dog is very sad.
She's be hiding. Oh, did you like?
I shut up like a while ago.
Okay.
It's okay.
Yeah, sorry about that. It might be in hiding. Oh.
No.
Surprise, surprise.
Yeah. Yeah. Really?
No, they were still testing. I'm signed in, I just haven't hit the start button.
But it doesn't matter who.
Oh, okay, great.
Just.
I should be able to. Might not.
Well, we're gonna have 4 dogs.
You should be able. It should be 5 in May.
My parents are.
There's a passcode. Did you?
Because Goose is like.
No, I haven't started anything yet.
But
So she kind
it
of-
should be good to go.
She's very cute.
Okay, great.
Yeah.
First thing in the morning, all she wants to do is play Kale. Kale's not. She's 6 a.m. None of the rest of us are ready for that kind of attack. So they get into this morning part. So we're trying to. I mean, she's gonna go right now. So it'll be good for spread the. Yeah, they're good. We're going to a wedding next week, so we're
Am I good to start biggest line?
fingers crossed. Yeah. It's time here. So.
No need to sign in if you have a name tag. Thank you.
I haven't registered.
Okay. Yeah, you can sign in
We tried to be
if you'd like. You can.
Emily's a good guy.
Name tag.
They get one of them's really capable or uses to like set her. That was right. Yeah.
You know,
You just talk.
I know.
I'm like, I'm kind of.
If you're quiet, you can.
Sure, yeah. Yeah.
Then you may want to use it,
Feel,
but if you can project and make
you
that.
know, put my arm
So
Hear you.
and I should left.
that's fine. If you want to make a new one, you can. Did you register? Okay, then go ahead and sign in if you'd like to make yourself. Go ahead.
It's crazy. She's
Remember my name for it?
yeah. Maybe it's just because she knows she's and just like you have and her family's over relatives. We can't even walk.
2 months
We
straight.
can't cross the
A month later, she goes,
You get a power suit, put it on. What are you doing?
Can you get Johnny?
We're done. Thanks for, doing all the organizations.
Oh, yeah, no problem.
well, it's very but if you get that
Recording in progress.
Benefit your future.
Today. Maybe we'll have a chance to. For the rest of the day. Easier to do it like in person.
No, we registered.
The one that's
So much nicer that kind of solve. Yeah.
no, I haven't actually. I was hoping.
Grab a little.
We're not gonna find it.
Sure. Yeah.
Olaf, I don't know if you remember.
Hey.
Yeah.
About this first.
I go say
Yeah.
Senior grad student.
Yeah, right. This should be
Yeah, yeah. Register. But you should have a name tag.
Seminar, right?
If you don't have a name, you
Walk on the street.
We are out there. Yeah,
You go with your
I don't know. I know that feeling. They're like
I'm busy.
for everything.
Last.
Yeah.
I almost can't do it.
Yeah.
Elbow bump.
Yeah.
Well, when did you start?
August.
There's
Yeah.
military. I don't think you did. Name. You'll have to make your name tag.
Yeah.
And a name tag of shame.
Yeah.
Okay.
Actually, you're right. I will totally write next time.
That was back then.
I've heard like a
Have a name tag, no need to register. You can just drink. You can just hold it like
That way, it's
yeah.
from the knowledge and everything. Like for it's just like up. So yeah, but something we're running to or seeing here.
Yeah, well, sorry, I
Yeah, you know, but that's some grants.
I knew you did this.
But certainly,
That was what you learned.
we're still getting yes.
I don't know. No, it's going. Well, I did it back.
Because I think
If you wouldn't mind, just
too late to add.
I'll let you know if I can cross it, but if I didn't, yeah.
Wait, did you carry over much of your weight?
I know it's Hi Warren. Got you from yesterday, yep.
One of the rhymes.
Yeah.
That one.
Yeah. I had a few of those like similar things that was like Pascual.
Okay, I think we're about ready to get started. You guys could take your seats.
I wonder if it showed up as you attending.
Oh, no.
So, welcome everyone to our second day of the Fall Symposium for the Center for Ecology and the Environment. I am Wendy Turner. I'm one of the co-organizers of the symposium with Carl. And thank you very much for showing up here today, those of you who made it in person and those of you who are here online. Welcome. We will jump straight into our excellent speakers today. Fire that up. Our first speaker today is Dr. Jess Hua. She's an associate professor in the Department of Forest and Wildlife Ecology, and she's a new addition to the UW-Madison community. She joined us this fall. She got her PhD at the University of Pittsburgh and did a postdoc at Purdue University before starting as an assistant professor at Binghamton University. Her research focus is on fundamental mechanisms shaping ecological interactions, evolutionary processes, and eco-evolutionary feedbacks in human-modified environments. She works in aquatic ecosystems, working on amphibians, ecotoxicology, and disease ecology. Dr. Hua has a thriving research program funded by NSF grants and various foundations. She incorporates outreach and community science into her research, collaborating with artists, educators, and engineers in her science communication. She's even co-authored a children's book on wetlands. So today, Dr. Hua will be presenting a talk titled Pollutants, Parasites, and People, Disease Ecology in the Face of Environmental Change. Thank you.
Thank you so much, Wendy, for the introduction, and thank you all for being here. I'm really excited to have the chance to share some of the work that we've been doing in my lab for the past few years. So we all recognize that the world is changing in many different ways. In my lab, we're interested in understanding how global change influences ecological interactions, and specifically the interaction between hosts and their parasites. So when it comes to host-parasite interactions, one approach might be to ask, well, what are factors that influence a host's ability to defend itself against parasites? Alternatively, what are factors that influence the parasite's ability to infect hosts? Building upon this simple interaction, the field of disease ecology has increasingly recognized the importance of the environment in shaping these interactions. In my lab, when it comes to the environment, we're interested in pollutants from things like pharmaceuticals to road salts, microplastics, light pollution, and we're interested in how this shapes host-parasite interactions. Now, while this disease triangle framework provides a really nice foundation for asking how pollutants influence these interactions, one thing to keep in mind is that they don't occur in a vacuum. For example, it's not just a single host, single parasite that is interacting, but often multiple hosts that are interacting with each other as well as with their parasites. And so a central theme in my lab is to ask, how can we integrate ecological complexity into our understanding of disease outcome? So the first part of this talk will focus on some of our work with road salts, and specifically how road salts influence simple host-parasite interactions. And then building upon this with a little bit more ecological complexity, how do road salts influence host-parasite interactions in multi-community systems? Now, we're interested in introducing this ecological complexity, but at the same time, also asking how can we build upon this disease triangle by integrating an evolutionary perspective. We're interested in asking how pollutants might cause selection on hosts, and ultimately, how this can then have an impact on host-parasite interactions. So, towards this end, we ask it in the simple system, single host, single parasite, and then we'll build it again by asking it in more complex systems. One challenge when it comes to adding ecological complexity is that oftentimes it's not enough for, it's too much for a single lab to do. So with that, I'll tell you a little bit how we have collaborated with citizens and community scientists to address these goals. So this is the outline for today's talk, and I will jump right in. I'll first tell you about our work with road salts and host-parasite interactions. In my lab, we work with 3 different types of amphibian parasites and amphibians as our hosts. For the sake of time, in this section, I'll focus just on the trematode. This is a very common parasite. It's free-swimming, and what it does is it infects larval amphibians. It will swim up the cloaca of tadpoles and encyst in the kidney. It is dose-dependent, typically not lethal. So does sodium chloride modify host-parasite interactions? I'll talk about some of our work with wood frogs and Echinostoma trivolvus, the trematode. This is also work done by my former PhD student, Nick Buss. He took wood frogs and exposed them to 4 different concentrations of salt. For reference, the highest concentration of chloride detected in wetlands in our area was about 4.6 grams per liter. However, the majority of wetlands have a much lower concentration. Here are our concentrations. They are environmentally relevant and sublethal to wood frogs. So using these concentrations, after exposing wood frogs, Nick exposed them to 50 trematodes and asked how many of these trematodes successfully encysted. So on this data slide, on the x-axis is sodium chloride concentration, on the y-axis is average number of cysts when not exposed to salt compared to when exposed to salt. For all sublethal concentrations, exposure to salt increased tadpole susceptibility to trematodes. So taking a step back, what are the take-homes here? And I think the important point is that in the field of toxicology, we often think a lot about what are the lethal effects. And I think studies like these underscore the importance of not just the lethal effects, but the environmentally relevant lower concentrations that might still have negative consequences by impacting the ecological interactions. Of course, as I alluded to before, one limitation of this study is just a single parasite, a single host. In reality, there are multiple hosts interacting with each other and also with their parasites. In fact, this host biodiversity disease relationship is a topic that has been hotly debated in the field of disease ecology. And why and what is the biodiversity disease relationship and why is it so interesting? It's the idea that biodiversity is linked to disease outcome. So a common pattern that people see is this dilution effect. As you increase in species richness, individual and overall community infection is reduced. And this is exciting because it provides a clear benefit for why we might protect biodiversity. You protect biodiversity, you get a benefit. The challenge, however, is that sometimes you get this, sometimes you get this, and sometimes you might even get this. And so where the field stands right now is, what are the mechanisms driving these shifts in relationships? And in the second section, what I'll talk about is one factor that we think might be driving the equivocal nature of this relationship. So before I do this, let me just walk everyone through the one mechanism driving the dilution effect. So we have two communities, lower host diversity, higher host diversity. In our lower host diversity, if we imagine we have hosts that are highly susceptible. If there are parasites in the water, these hosts have a high chance of getting infected. In contrast, in a higher host diversity community, theory predicts that we're much more likely to have hosts that have that are less susceptible to the parasite. In this situation, you have parasites, and these less susceptible hosts may draw the parasites towards them, but these parasites are unsuccessful at infecting them. So these tadpole hosts that are less susceptible can act as decoy that can reduce overall infection. So it looks something like this. The challenge, however, is that many of these studies often occur in benign environments. This is important because in that last section, I just showed you that a pollutant like salt can modify patterns of susceptibility. So these decoy hosts, in the absence of salt, may be great decoys, but in the presence of salt, they may have increased susceptibility, and even though you have higher host diversity, you may not get that benefit of the dilution effect. So we hypothesized that pollutants can influence the magnitude of the biodiversity disease relationship by modifying patterns of disease susceptibility. To address this, we had to figure out two things. First, we needed to understand what does salt do to different host species. And once we have this foundational information, then we can ask what are the cascading consequences on that relationship. And again, this is work done by Nick Boss. So to address that first question, Nick repeated that same experiment from before, except with different species and fewer salt concentrations, exposed them to trematodes and counted how many encysted. On the x-axis here, these are the host species. Y-axis, average number of cysts per community, and I'll present data on just communities without salt. So the main take-home here is that it doesn't matter the host species, they all have similar levels of susceptibility in the absence of salt. In the presence of salt, we found that peepers became more susceptible to the parasite, wood frogs also became more susceptible, and toads did not change. So now that we have this foundational information, we can then develop testable predictions about what we might expect to happen with that relationship between biodiversity and disease. So in the absence of salt, all three species have similar susceptibilities. So as you increase in species diversity, there isn't any one species that can act as that decoy. So we predicted that there wouldn't be a real benefit to increasing species diversity. There isn't a one species that is less susceptible drawing those parasites away. In contrast, in the presence of salt, what we find is that toads are significantly less susceptible. So in this situation, as you increase in species diversity, we predicted that any community with the toad would have a reduction in disease susceptibility. So how did we do this? We generated a number of different communities that differed in the number of species, though we kept the number of individuals the same. We raised them in salt or no salt environments, exposed them to the parasite, and counted how many successfully encysted. So on this data slide, let me just first orient everyone to the x-axis. From left to right, it goes from lower host diversity to higher host diversity. In the first panel, it's all of these different diversity communities compared back to the peepers, here compared back to the wood frogs, here compared back to the toads. As a reminder, all three species had similar levels of susceptibility, so we did not expect increasing diversity would benefit them by reducing their parasite load. So I won't spend too much time on this slide other than to show that we, as expected, sorry, this is a little bit slower, did not find evidence of a dilution effect. No dilution effect. So in the absence of salt, we found no dilution effect. In the presence of salt, as a reminder, we get a shift in the susceptibility. Peepers and wood frogs become more susceptible relative to the toads. So the key is that in the presence of salt, pollutants modify patterns of host susceptibility. How does this affect the relationship between diversity and disease? Again, peepers and wood frogs were highly susceptible to the parasite. Toads were not, so we predicted that any community with a toad would have a reduction in disease susceptibility. For the most diverse community, we predicted, again, a reduction. However, because in this community there are only 2 diluter species to 4 susceptible species, we predicted that the decrease wouldn't be quite as high in magnitude. So relative to the peeper-only community, the peeper plush toads, we found a reduction in the number of cysts per community. Similarly, compared to the wood frog-only community, we see a reduction in the number of cysts in communities with toads and wood frogs. And then for the most diverse community compared to peepers, we see a significant reduction. For the comparison between the most diverse and the wood frogs, we see a decrease, but it's not significant. So I fully recognize that I just put a lot of data on there, so I'm going to summarize it in this slide. We found, first, sodium chloride exposure altered host susceptibility to parasites. By altering host susceptibility to parasites, we found that this had cascading consequences on the direction and the prevalence of the biodiversity disease relationship, specifically the dilution effect. So this work is done on road salts. We know that there are a number of different pollutants in the environment that can do very different things to susceptibility. And so the main take-home that I'd like to leave you all with is, again, we're seeing these very different relationships between biodiversity and disease, and I hope I've been able to show you that one factor that might be contributing to these shifts in relationships might be the effect of pollutants on patterns of host susceptibility. So I hope this came across clearly because this will come back again in the final part of this talk. In the meantime, I'm going to shift gears from talking about the ecological perspective to talking about the evolutionary perspectives. Specifically, in this section, I'll tell you a little bit more about our work on how evolutionary responses to pesticides can influence host-parasite interactions. So when it comes to the evolutionary responses to pesticides, typically what people think about is this idea of genetic changes in constitutive existing traits due to natural selection. You have a population, a pesticide comes through, some individuals have an existing constitutive ability to deal with the pollutant. If it's heritable over time, you get the evolution of tolerance in that population that allows it to persist. One challenge, however, with this mechanism of tolerance is that it can take multiple generations. This is a challenge because oftentimes when pollutants enter into the environment, it's rapid. So we became really interested in thinking, are there other mechanisms that might allow organisms, wildlife, to more rapidly respond to pollutants in their environment? And this led us to thinking about phenotypic plasticity. So in this picture, you have tadpoles that are highly plastic, responding to cues in their environment. These tadpoles are from the same egg mass. The only difference is that tadpole on the bottom was raised in an environment with a predator where it, within its lifetime, induced an adaptive trait that allowed it to better respond to that predator environment. So we became curious if we could do it with a predator cue. What about a non-lethal pesticide cue? In other words, can tadpoles within its lifetime induce tolerance to pesticides through phenotypic plasticity, in addition to constitutive tolerance? Before I get to those data, let me just introduce the reaction arm figure that I'll be using. On the x-axis is the environment, no pesticide or pesticide. Y-axis is pesticide tolerance. So if a tadpole were plastic, in an environment with no pesticides, we might expect low tolerance. However, the same tadpole, if raised in an environment with pesticides, would have the ability to induce increased tolerance. So a sloped reaction norm line indicates plasticity. So we thought we might increase our chances of finding this, so we turned to theory. Where in nature might we find plastic individuals? Where in nature might we find constitutive individuals? So we started thinking about genetic assimilation theory. Plasticity should be favored in environments that are variable. If environments are variable, it makes sense to maintain the machinery to detect and respond adaptively. So what do I mean by variable pesticide environments? Things like wooded ponds where you might have drift of pesticides here every once in a while. Now, theory predicts that as you move towards consistent pesticide environments, plasticity is no longer beneficial. In fact, it can be costly to maintain the machinery to respond, to detect and to respond. So in a pond that might be next to agriculture where pesticides are always present, you might see the loss of plasticity in favor of constitutive tolerance. So you go from plasticity in areas away from agriculture to constitutive tolerance in areas close to agriculture. And this is the prediction that we tested. So we have this same x and y axis here. Here are the different dotted lines represent different populations. We find populations that are inducible to pesticides, and these are populations that are far from agriculture. As you move closer and closer to agriculture, you see that average reaction norm line flattening out, and you see that shift from inducible to constitutive tolerance. So we were really excited to find that these results were consistent with genetic assimilation, but the real reason that I'm showing you this today is because this gradient provides us a really useful framework to ask this broad question. How does selection by pesticides influence disease outcome? We have these different evolutionary strategies, plasticity and constitutive tolerance, that might be associated with differential costs or benefits. How might these then have cascading consequences on disease outcome? And this is the work of my PhD student, Vanessa Werthner. She went out to these populations that differed in plasticity or constitutive tolerance, raised them in common garden environments, and then exposed them to parasites. Again, for the sake of time, I'll focus on a different parasite this time. This is frog virus 3. It's an emerging infectious disease that is very lethal. It is transmitted via contact, so it can cause mortality within about 7 days. So what did she find? On the x-axis, this is it moves from plasticity to constitutive. Y-axis is ranavirus load. The data here represent only survivors. This is important because the y-axis can be a little bit counterintuitive. The higher ranaviral load actually means they're more tolerant because they were able to survive with higher loads. So Vanessa found that there is a relationship between evolutionary tolerance, or the mechanism for tolerance, so whether they're plastic or constitutive, and specifically she found that populations with constitutive tolerance were more tolerant of, more ranavirus. So from here, this was work that we found in 2017. For the past few years, we've really begun to dive deeply into why this might be. Why are plastic populations different than constitutive populations in their susceptibility? We've looked at things like oxidative stress, corticosterone, microbiota differences between these populations, behavioral differences. I don't have time to talk about all of these things, so I'll give just a quick snapshot with behavior. And we care about behavior because, as I mentioned before, this pathogen is transmitted via contact. And in particular, contact through necrophagy, which is the consumption of dead conspecifics. So an undergraduate in my lab was really interested in pursuing this and asked, do tadpoles with constitutive tolerance behave differently than tadpoles with inducible tolerance? What she did was she took tadpoles, exposed them to the virus. Once they were infected, she generated two types of communities. Communities with only inducible tadpoles and communities with only constitutive tadpoles. And she introduced these infected tadpoles, and what she measured was necrophagy, viral load, and final survival. So I'll focus on just these two response variables today. So you'll see two pie charts coming up, and for the plastic populations, what Mary found is that the majority of the tadpoles with plasticity to pesticides exhibited this necrophagy behavior. In contrast, for the tadpoles that had constitutive tolerance to pesticides, only 11% exhibited this behavior. So how does this translate to disease outcome? So the plastic individuals exhibited more necrophagy and also survived more poorly compared to those that exhibited constitutive tolerance to pesticides. And so what this suggests is that behavioral differences between plastic and constitutive individuals might be a promising direction for us to continue to follow up on in understanding what are the mechanisms driving these relationships between the evolutionary strategies for tolerance and disease susceptibility. So in short, I can tell you that we've pretty much knocked out oxidative stress and corticosterone. Those were not related. there's some promising microbiota data. The tadpoles that were more tolerant to pesticides and more tolerant to FE3 also had higher microbiota diversity. We're interested in the microbiota because the diversity of the skin microbiota often is that first line of defense for these tadpoles. So that brings me to the end of this individual level section. In the last 3 minutes or so, let me tell you how we have worked to increase in complexity. We've done this, as I mentioned before, this is coming up again, this biodiversity-disease relationship. As a reminder, we showed that pollutant exposure altered host susceptibility to parasites, and this shift in host susceptibility modified the biodiversity-disease relationship. I just showed you that tadpoles with inducible or constitutive tolerance to pesticides influences their susceptibility to ranavirus. Where we're going next is, does pesticide tolerance strategy also, is that another factor that can shape this biodiversity-disease relationship? Is it another cryptic factor that can shape why sometimes you see dilution effect and sometimes we don't? To address this, we'll do lab studies, manipulative and highly controlled studies, but of course a limitation of that is that how does this translate to nature? And so to do this, we've been working with community scientists to help us collect data in the field. And when we're working with community scientists, there are a few questions that are important to us is, how can we generate community interest in research? Of course, I love it. My lab loves it. But can we get people to care about the research that we do? And then also, how can we ensure high-quality data collection? So to address that first question, we've been working a lot with art and how we can use art to improve public interest in research. This is work done by Benjamin McLaughlin, my former undergraduate student. And what he did was he took several wetland research papers and translated them into an art show. So he created this art show, and then he invited university community members, so mostly students, to come see the art show, and we assessed it doing pre and post surveys. There were 188 total participants, and what Ben was able to do is he was able to break them up into different majors. So what did Ben find on the x-axis? Before exhibit, after exhibit, y-axis, interest in research scores. For those students that were non-STEM majors, the art show really made a difference. After going to the art show, there was a significant increase in research scores. For the general STEM students, there was an increase, but it wasn't quite as dramatic as those from non-STEM. And for the ecology-specific individuals, they didn't change at all. They already had a high interest. So I think what this suggests is that the art show can level the playing field in terms of interest for students that are non-STEM. So art can improve public interest in our research. We're really excited about that. How can we use this information to ensure high-quality data collection? So we developed a 3-step process. 1, content development using art. 2, using this content to teach teachers and their students how to help us collect data. And then data collection. How does this look in real life? Here's some content we generated using graphic novels to teach 3rd graders about amphibian biodiversity and the risks that they face, paired with a lesson plan. These are the incredible undergraduates that worked on this. Then, using these resources, we trained teachers how to teach students about biodiversity and disease. They were paid to come to our workshop. Huge incentive, I think, for them. And they also got teacher training credit. Then they taught their students. And then we assessed perception of science, how this activity changed this, and also science content. And then we bused 3rd graders to a local museum where they helped us to collect biodiversity and disease data. Where are we now? We've gone through 26 classrooms with over 500 students. They go to different locations that differ in human impact. They help us ID the biodiversity of those locations. This is the axis that they're working on. Right now, we're working on overall infection. This is being done by first-year research immersion students. So they are quantifying disease in the same tadpoles that the 3rd graders helped us to identify. And they will also be going to the ponds to quantify human impact by measuring water quality. So that brings me to the end of this section. Who are we? We're the Whole Lab. We are interested in pollutants and their effect on the environment and host-parasite interactions. We are working to integrate in ecological complexity to that understanding and also add in that evolutionary perspective and also linking eco-evo dynamics. We're really, really passionate about science communication and outreach and incorporating the community to help them understand the scientific process. The funding sources that we are so grateful for, but I can't leave without spending time. This is my lab. They're amazing. They are the ones that did all of this work. I just get the opportunity to come up here and present. So with that, if there is time, I'd be happy to take any questions, and thank you so much for y'all's attention. Yes.
I was just wondering, what were the mechanisms that the frogs were using to resist the parasites? Were they using slime, or what was their immune function doing?
So there are several hypotheses for what it is that the amphibians are responding, or their immune system for coronavirus specifically. One is the microbiota. Many of the microbes on the microbiota create supernatants that are antipathogenic. So that's one hypothesis for why that might be the first line of defense. Another are shifts in behavior. So the shift in behavior in necrophagy or the avoidance of infected conspecifics. But I think there's no real consensus in the literature as to what the mechanism for tolerance is yet for this particular emerging infectious disease.
And what was the pesticide that you used?
We use Carbro in this particular study, but we also use a wide range of other ones, from the organophosphates to pyrethroids, neonicotinoids. So we've done the history of pesticides from organochlorines all the way up to the more recent ones. Mm-hmm. Yes.
Thanks for a really interesting talk. I had a couple of questions. The first one was, I noticed that your depictions of the tadpoles show them different sizes, and I was wondering if there's an element of like biomass and pest load that could help explain some of those effects. And then the second question was about the necrophagy.
Mm-hmm.
Like, what fraction of their diet does that normally represent? Is that a big cost to be missing out on it, or is it like just a tasty snack?
Okay, I'll start with the first question. The different sizes, yes, absolutely. We know that variation in stage, so development, and also size can influence specifically the trematode susceptibility. So larger individuals can hold more parasites. So how does that affect the biodiversity disease relationship? I think that in many ways, because pollutants can also change size. And we've looked at this across the wide range of pollutants that we've looked at, because one of the questions is, like, you increase in complexity, how much in complexity do you do? And so we started looking at some of the generalizability across these pollutants. Sometimes they increase disease susceptibility, sometimes they don't. But the one factor that we can that seems to be consistent is, if a pollutant changes mass by making them larger, then that's when we see increases in disease susceptibility. So yes, we're starting to think a little bit more about adding that level of complexity, but I think we don't know quite so much how the interaction between size yet and pollutants, like how the specific mechanisms for that, but we're working on it. Your second question was about
the fraction of diet that necrophagy usually constitutes.
I do not know the answer to that. I do know that these tadpoles are very opportunistic. For the most part, if there's algae, they prefer algae. But in highly competitive environments, I don't think that a resource is a resource, at least from the communities that we have worked with.
I think there's one question from online.
We do have one question from online. Do pesticides or other pollutants affect the immune system that affects survival?
Do pesticides affect the immune system? So not necessarily. We haven't looked at the immune system specifically, but we have measured corticosterone and other physiological metrics like oxidative stress. We have found that pesticides can reduce corticosterone in certain situations. How that translates to increased disease susceptibility, we're not sure. One mechanism is that by reducing corticosterone, it may make tadpoles more vulnerable by reducing their ability to respond to the pathogen. In terms of oxidative stress, we found actually not much in terms of shifts. Some increases in factors that can protect amphibians from oxidative stress, but as far as we have looked, not much there quite yet. But we are just now starting to get into that, so I wouldn't say that we have enough information to say yes or no.
Okay, well, thank you very much, Dr. Bowe.
Thank you.
Okay. See how much multitasking I can do. Okay, our next speaker today is Dr. Olaf Jensen. He's an associate professor in the Center for Limnology. You don't need to use the mic if you don't want. There is a mic here. If you think you can project, it's your choice.
Sounds good.
Okay, sorry to interrupt that. So Dr. Jensen has rejoined the UW-Madison community as part of our pandemic cohort of recent hires. He came in 2020, I believe. But he did his PhD and a postdoc here, and followed by a Smith Conservation Fellow at the University of Washington. And then he started as an assistant and then as an associate professor at Rutgers University. So Dr. Jensen has a really productive and impressive research program focused on fisheries and aquatic ecosystems, including marine, estuarine, it's a very hard word to say, estuarine, and freshwater environments. Dr. Jensen's study systems range from field studies on endangered salmonids in Mongolia to meta-analyses of stock assessment data to better understand fish population dynamics. And today he'll be speaking about Are Marine Fisheries Sustainable? Comparative Analyses of the World's Largest Fisheries. Thank you.
Thank you for the introduction, and thank you for the invitation. That's a tough act to follow. That was a fantastic talk. I really enjoyed that. So today I'm going to talk about a variety of work in my lab through postdocs and graduate students in my lab, as well as work with a broader set of collaborators, many of them at the University of Washington. A lot of the work was funded through NC's working groups, NC's. National Center for Ecological Analysis and Synthesis. Great. And all of it framed toward answering various aspects of the question of whether marine fisheries are sustainable. And like any ecological question, the answer, of course, is it depends, but we'll go a little bit beyond that, hopefully. So the basic problem is that world fishery landings peaked all the way back in the 1980s. That's total wild capture fisheries production globally. And of course, the human population has continued to soar way past that, leaving a gap here between demand for protein and the available supply from wild capture marine fisheries. Aquaculture has filled a lot of that gap, but I'm not going to talk about aquaculture today. It's really a very separate set of research questions, much more controlled, much more along the lines of agricultural research questions. So we'll take a look at that issue of the difference between supply and demand and ask whether we might be able to make up some of that by fishing harder. That is, are we sort of underexploited on a lot of fisheries, or are we better off letting overfished fisheries recover? Of course, if you pay attention at all to the media, this question is already answered, and none of this is necessary because we all know there are oceans of nothing. And so we decided to try to take a little bit more quantitative approach to this. And I think to get there, it helps to think of fisheries as kind of a weird retirement account, right? So you've got some principal in blue here and some interest. And the idea is you want to be able to live forever off of the interest without a long-term decline in the principal of your account there. And of course, when the principal is small, when the stock sizes are low, this interest, the surplus that they generate is small as well. But unlike a retirement account, the interest, the surplus is small when the population size gets high as well. And that, of course, is just the ecological carrying capacity. Once you're up near carrying capacity, the potential for growth is low. And so the relationship between that interest, or surplus production as it's called in fisheries terminology. Fisheries has an awful lot of arcane terminology. That surplus production is dome-shaped. When you're up near carrying capacity, there's no surplus production, and of course, if you've driven them down to extinction, there's no surplus production as well. And this works for stock size, the sort of biomass of the fish population as a whole, or the fishing rate. Either of those should have some intermediate value that gives you your maximum sustainable yield. And so that's just denoted from BMSY, the biomass associated with that maximum sustainable yield, or UMSY, which is the fishing rate, the fraction of the population that we harvest each year associated with maximum sustainable yield. So to get there, we compiled a global database of stock assessments. And stock assessments are essentially the information, the mathematical models applied to time series data that are used to inform fishery management around the world. And these include time series of biomass, that's a model estimate, catch, what's taken out of the ocean, the fishing rate or fishing mortality, and this surplus production, the extra that we can harvest every year without long-term declines in the population. As well as things like reference points, so what the fishery managers are targeting, what they're trying to achieve, and management characteristics and some life history information about the fish and invertebrates that are included here. And that's all a publicly available database. We talk about it as global, and certainly in terms of the extent, it covers much of the world. It only covers about half of world fisheries catch. There is another half of world catch largely taken from the tropics and from East Asia, where the coverage is particularly poor here. I'm not going to try to say anything quantitative about the status of the other half of world catch, other than to note that everything we know about that other half says it's nowhere near as sustainable as these large industrial fisheries that are represented in this database. That's a major caveat. So first of all, where are we relative to these two reference points? So we've got this relative fishing pressure here on the y-axis, and that relative biomass on the x-axis. If we are fishing exactly to the reference points, we'd be right here in the middle of these crosshairs. So that's sort of where you would want to be if you didn't build in any buffers for precaution. And the various quadrants here, you can think of this one up here as sort of the emergency room. So biomass is below the target, and we're fishing higher than we're targeting, right? Higher than we know is sustainable. This is the recovery room. This quadrant here should not be one in which stocks spend a whole lot of time. It's a non-equilibrium state where the biomass is low, but the fishing pressure is also low. So these stocks, if the models are right, it's a big if, should be moving in this direction and recovering. This is kind of the happy place, at least from a conservation perspective, where we're not fishing as hard as we could, and the biomass is higher than it would be otherwise. And finally, this is sort of the Wile E. Coyote quadrant, where we've kind of gone off the edge of the cliff and don't know it yet. Fishing mortality rates are high here, but the biomass has not yet declined. This is another non-equilibrium condition. It should be moving to the left. So where do real populations fall out? All over the place, largely not in this non-equilibrium quadrant up here. Don't worry for now about the difference between the blue and the gold ones. It's just different ways of estimating where the populations fall here. And I think there's good news and bad news in this graphic. The good news is that almost half of populations fall into this quadrant over here, representing a little bit more than half of the catch from the database here. So about a quarter of global catch comes from that sustainable quadrant over here. Another quarter there of the assessed stocks are in the emergency room still, so not good. Biomass is low and fishing mortality rates are still high. And slightly less in the lower left quadrant there, where historically, presumably, the fishing mortality rates were high enough to drive the biomass to low levels, but are not currently too high. So these stocks should be recovering through time. And in fact, recovery through time is what we see when we take a time series look at global fishery status. So the red here is the biomass. This is how hard we're fishing in green here. And this is the absolute catch, which tracks very well how hard we're fishing. And what we see here is some real changes in time here, right? There's this initial phase up until the mid-1990s when biomass was declining. We were fishing increasingly hard. And after the mid-1990s, there's sort of an inflection point here where the biomass starts to recover after the fishing mortality rates in there. So just as we would expect from the models, once we start to ease off the accelerator here with fishing, the population biomass globally started to recover. So that's the average story, but it's hard to look at this and say that there's some kind of clean average story. In fact, there's mostly just a whole lot of variation here. And so the obvious question is why? So why do we see so much variation in stock status across fisheries around the world? Well, as a good ecologist, a card-carrying ecologist, I think my card must be in the mail still. The fish life history was one of the first things we looked at. You know, are there life history differences between these populations that are doing well and poorly? Particularly, you know, are these slow-growing, long-lived species? Are they the ones that are in the worst shape? We looked then at critical population thresholds. Are there differences in sort of the point of no return in different populations that can explain these differences? Have some of them just been pushed beyond a point of no return that is a little bit closer to the targets than others? And then climate change, of course. How much can we explain of the variation in population dynamics and status from change? I'll start with the first one. From stories, from narratives about fisheries, this one seems obvious, right? We can think of big fish like the bluefin tuna, long-lived fish like many sharks and rays, or cod, another big fish that all of these are quite depleted in many populations around the world. So is it these slow life histories that predispose fish populations to depletion and eventually collapse? Well, we looked at this using a variety of life history traits drawn from a separate global database of fish life history characteristics, FishBase, and divided these 5 metrics into sort of fast and slow variables. So we've got longevity, we've got short-lived and long-lived species, age at maturity, maximum weight, trophic level, which doesn't quite follow along here, but tracks a bit with maximum size. And there's this other hypothesis out there, fishing down the food web, which probably you've heard of, which posits that these higher trophic level species get depleted first. And then growth rate. So how did it turn out? There's nothing there. There's nothing there at all with respect to these life history traits. If anything, it's slightly in the opposite direction, where the faster life histories have a higher probability of having been collapsed at some point in the stock's history. But we're talking about really marginal effects here that explain almost none of the variation. So it's not life history, not in any predictable way anyway. We'll come back to that. What about the idea of critical population thresholds or so-called strong allele effects? The idea here would be that that if you plot sort of the number of spawning adults and the surviving offspring, that there's a relationship like this typically, but where this is an equilibrium point, the dashed line here is just replacement. And so where replacement crosses that line, you get a stable equilibrium point, right? In the absence of fishing, everything should kind of fluctuate around that stable equilibrium point. Well, if you have a Lee effects, there's a curvature to this, and it crosses the line again down here, and you should get a non-stable equilibrium point down there, another critical point where below that you tend toward extinction. So stocks that are too depleted might never recover. So a former postdoc of mine, Phil Neubauer, led an analysis of this using the RAM legacy database, looking at 153 stocks from that database, which at some point had dropped below half of their target biomass. Half of the target biomass would be typically a level associated with implementing a recovery plan. Some kind of emergency action would usually be triggered at that level, at least in the US. Other countries have similar sort of emergency thresholds for declaring a fishery overfished. So he looked at that using a Bayesian survival analysis. This is essentially a quantitative technique from the medical literature looking at treatments, in this case changing the fishing pressure on fish stocks and asking them how they respond with an open-ended outcome. We don't know the eventual outcome for all of these because we're still fishing these populations. And he asked whether depleted fish populations recover, and if so, how long it takes. So the model included the potential for an infinite recovery period, essentially non-recovery in some of the stocks. That option was not supported almost across the board, but recovery can take a long time. So depending on the intrinsic rate of increase of the population and how far they're depleted, recovery, even in the absence of fishing, might take 40 years or more. So to some extent, when we saw those populations clustered in the lower left-hand corner there, the ones that are at low biomass but are not being fished very heavily, we might expect them to be there for some time if they were driven to very low levels, and especially if they're populations with low intrinsic rates of increase. Okay, and finally, moving on to the climate change hypothesis. This is work done by a former PhD student, Chris Free, at Rutgers, looking at whether there are climate change winners and losers. You know, does warming in the oceans increase or decrease productivity, and how that changes overall potential for sustainable harvest from the oceans. So what he did was to take that simple dome-shaped relationship that I talked about before and make it dynamic and a function of temperature. So for a climate change winner like black sea bass, in years when temperatures are higher, these reddish points, the productivity is correspondingly higher as well. And the cooler years, the blue points down here, productivity is reduced. This is one of the clearer examples of a winner from climate change. Similarly, there are climate losers, and this is one of the clearest of those, Atlantic cod in the Irish Sea, where all of the good years, all of the years with high relative productivity are colder years, and the years with warmer temperatures are all lower productivity years. Most populations are climate indifferent, or at least too noisy to tell is probably a more accurate answer there. So he did this across all of the stocks in the RAM legacy database for which sufficient data were available, and was able to take them and plot them out essentially on this axis here with this temperature influence parameter theta ranging from negative values, the climate change losers, to positive values, the winners like black sea bass. And what he found was that in the final model, roughly balanced. So 19 stocks negatively influenced, 9 stocks positively influenced, and a whole lot of stocks in the middle that didn't have very clear signals there. However, this looks like just noise. But when he ran the null model, which randomized all of the data here, it turns out quite differently. Essentially, there's no climate effect, and that's easily distinguishable from the final model there. So roughly balanced, that's great, but it depends where you are. So across the world, there are regions with strongly negative impacts of warming and others with positive impacts. And worrisomely, one of the areas with the strongest negative impacts is over here in East Asia, where reliance on seafood for protein sources is highest. Also, Northern Europe here, another area of fairly negative effects of temperature overall on average. These don't match up well with areas of good or poor stock status. And part of the reason is that when you add it all up, there's a rough balancing here between the benefits from warming for some of the stocks and the detriment of warming for others. If we look at the change in maximum sustainable yield added up over all of these stocks through time from the 1930s to the 2010s, essentially we see fairly a lot of interannual variation, but little long-term trend. The difference ends up being about 4%. So climate change has not yet driven huge changes in sustainability of marine fish populations. There are almost as many winners as losers so far, and the net effect when we weight according to the size of the populations is essentially almost a wash at this point. Okay, so it's not life history, it's not ELE effects, it's not climate yet. What is it? Well, there's substantial variation in sustainability in fisheries in space and time. So we looked at the temporal dimension, but we can look at different regions here and do the same pattern. The US West Coast and New Zealand are among those places that have similar patterns to that global trend, right? Where we saw the biomass decreasing up until the mid-1990s or so, at which point the fishing rates go down and biomass recovers a bit. Not so much here, quite a bit on the US West Coast. And then we've got other areas of the world that follow very different patterns. So the east coast of Canada here goes down and stays down, and it's even worse in the Mediterranean here and Black Sea, where we haven't even started to reduce the fishing mortality rates in any significant way. And so those populations obviously show no signs of recovery. So we've got a lot of variation among places, and we've got a lot of variation through time. If we dig into one country where we have pretty good data, the US, and ask about management, now we start to see something. So this is that same biomass trend for all US stocks. This is just the median biomass over the maximum sustainable yield biomass. That's kind of the target. And the Magnuson-Stevens Act is the legislation that governs federal fisheries in the US. The Magnuson-Stevens Act was originally passed in 1976, and you'd be hard-pressed to see any change in the trend at that point, right? That's not surprising. The original Magnuson-Stevens Act was not about conservation at all. It was about kicking out the Russians from US waters and taking those fish ourselves. Conservation wasn't even part of the equation at that point. However, the 1996 reauthorization coincides with a real inflection point here in stock status in the US. In 1996, there was a requirement instituted in the legislation that all fisheries have annual catch limits, that those be based on science, and that those scientific decisions about what those catch limits should be made by scientists, not the industry or fishery managers. So a real separation of scientific decisions and risk-based policy decisions, and a requirement to set hard limits. Not long after that, it was tested in the courts. The fishery management councils decided, this is really inconvenient to reduce fishing this much, and the courts upheld it. And since then, there hasn't been a lot of boundary pushing with respect to those limits. Another reauthorization in 2006 added some additional factors, but doesn't seem to have changed the overall trend of recovery very much. So that's the US. But a similar thing was happening globally around the same time. I'm not going to get into all of the different management measures implemented here, other than to say that the mid-1990s and early 2000s were really a time of very rapid increase in fishery management intensity around the world. A lot of the things that we think of as part of a modern sustainability-focused management system really started taking off around that time. Scientific surveys often predated those changes, but things like fleet-wide catch limits and harvest control rules that set really rigid limits on fishing all started to increase around the world around that same time. I'm not going to show it, but we also did a time series intervention analysis associated with this to look at when those different measures were implemented, what is the response of the fish biomass time series? And many of these have very significant effects on fish biomass. If we do the snapshot approach again and look at management intensity kind of summed overall versus this stock status in terms of biomass, apart from the tunas, there's a pretty good relationship. Rejecting the tunas there is not an arbitrary decision. Tuna fisheries are by and large essentially unmanaged in sort of the same, which seems odd. I mean, there's an enormous management structure around tuna fisheries, but they're managed in sort of the same way that a 100-mile-an-hour speed limit on University Ave here would be restricting, right? There's lots of other factors that come into play to restrict fishing before those management measures have any bite. In particular, the cost of going out and fishing in the open ocean for a widely dispersed creature like a tuna is so high that it really limits how much fishing can take place. So even though management intensity is fairly low for the tunas, their status is not too bad, simply because it's usually, for most of them, too expensive to fish them to really unsustainable levels. Okay, so to summarize, sustainability of large commercial fisheries in developed countries that do stock assessment, that is the places where we have the data, has greatly improved over the past couple decades, especially since about the mid-1990s. There's no secret sauce or ecological explanation. The places that have had the most recovery are places that have defined overfishing, made it illegal, and monitored to ensure that fishing actually stays within those limits. Climate change, I don't want to leave you with too much hope. Climate change impacts on marine fishery productivity have, on average, been modest and balanced between winners and losers. That's all looking backwards to the past, right? And asking how previous climate changes have impacted the historical performance of fisheries. We know that for any of these populations, even the winners, if you push too far in the warming direction, they'll go over the top of at least for growth, somatic growth of the body, a dome-shaped relationship between temperature and growth. They can be pushed too far, of course. So far, it's just that we haven't pushed that far. That's, of course, changing. So I'll stop there, and I'm happy to take any questions.
Beautiful overview and presentation. I especially appreciated your thoroughness of looking at all these potential factors and ruling out so many of them. But two that you did not mention come to mind. One is habitat structure, in particular these trawlers that have destroyed benthic habitats could be playing a big role. And the second is food web, trophic structure. Is there any measure of deviation from ancestral or pre-development food web structure that would allow you to assess that factor?
Yeah, so those are both two really, really good potential explanations. We have done some work on bottom-up effects of the food web, particularly if you harvest forage fish, do you see declines in productivity of their predators? And the answer, sort of surprisingly, is no. It's almost impossible to detect effects on the productivity of the predator from changes in the biomass of one or two of their species that they eat.
Deviation and top fed.
Deviation. Oh, it's sort of top-down control. We haven't looked at that. Potentially something to get into. I think we would have the data to get there. And then the other one you talked about was habitat modification. Certainly on continental shelf fisheries, many of these are continental shelf fisheries, impacts from trawling on the shelf can be large. However, the recovery times are actually quite fast as well of that habitat for the soft sediment habitats. So the soft sediment habitats, not rocky reefs or corals, and by and large nobody thinks trawling on corals is a good idea. But the soft sediment habitats are relatively dynamic and come back quite quickly. There's not a really good analysis linking productivity to that habitat change though.
But the Georges Bank has been very slow.
It certainly has. That doesn't seem to be due to habitat modification. The answer is really not clear there yet. Climate seems to play a role. In just north of there, seal predation seems to play a pretty significant role as well. One of the only exponentially increasing populations for decades has been the Cape Sable Island gray seal population. And I was recently on an examining committee for a student looking at that and figuring out how much consumption that population has, and it's enormous. It's enormous. So not necessarily for Georges Bank, but for areas north of there, the southern Gulf.
In the interest of time, I'm going to push any more questions to the social times after. Thank you very much. So before I introduce the speaker for the final talk today, I just want to present to those of you who might be new to the Center for Ecology and Environment Symposium series. What they do is they offer the keynote speaker an opportunity to give two presentations. So one, a more traditional research presentation focused on a topic of their choice, and then a second one, an opportunity to talk about another topic relevant to that that's close to their heart. And we've had presentations about outreach, about education, about graduate student mental health, about service, communication. So it's been a really broad swath of interesting topics. And in that vein, I'm really excited to present our speaker, Dr. Mercedes Pascual, for her second keynote event. So she did present yesterday and has been introduced more thoroughly. So I won't go into great details, and I'll be quite enthusiastic to jump into that. And let me just make sure I'm opening up the right talk.
Not the one from
correct. And let me get the slideshow. Do I need to open up the Okay, so I just wanted to say, again, if you missed yesterday, she's coming to us from the University of Chicago, where she is the Lewis Block Professor of Ecology and Evolution. She has received a great many number of awards and accolades for her very interesting work modeling and doing theory in ecology and epidemiology. And as a student of this field myself, I have to say that I found her work really foundational, linking
it's going on by
it's going on. No, I'm not supposed to be. Linking how climate variability links to dynamics of infectious diseases. So I think this talk will be really exciting, and I'm really pleased to welcome you. Thank you so much.
Thank you so much. Coming again, some of you, there are new faces, and I didn't get the part about close to my heart, so I'm not sure this topic is close to my heart, but I got the instructions that I couldn't really give you a research seminar. So there is a bit of research, but is sort of hidden under this topic. And it's something that indeed Jonathan Paz has seen develop. So I chose to talk about something that puzzles me, like why is it, and a bit for discussion, why is it that some polarized debates end up being productive and some ones you could have done without and have been a great waste of time in matters where we don't have time. And I don't have the answers. Some of the answers are actually evident, you will see, but it took 10 years to realize. And I should sort of say this is not meant as a criticism of specific scientists. Please take it that way. I really want to see what we can learn from that, because it was a frustrating period for me, but it's interesting that there are debates that have been productive and others that haven't. And why is it, in a world where we pose questions as alternative hypotheses, that this happens? And so I like to start with a quote on the grandfather of Darwin, which you can read, but he was a great philosopher in the 1800s, physician, etc. He was convinced that climate had a profound influence on human health. And you can say, for many infectious diseases, that is sensible. You get to modern times. This is, well, not so modern. This is an editorial commentary in Nature about essentially posing this opposed view: global warming versus intervention and socioeconomic development. The picture is that basically malaria may not rise because we have development offsetting the effect of climate change. And if you read between here in detail, it was this is a commentary on a published map of malaria, and we get to this later, but it was a reaction. So this field has gone like a pendulum, which is not profitable, from one side to the other. This was a reaction to earlier analysis that painted a dire picture of malaria-ridden future, blah, blah, blah. But in fact, if you read those earlier papers, there was discussion of the conditions. So I'll return to this because I see these kinds of editorials, the titles sell, but the titles are simplistic. And I think they do not help when there is not enough discussion of what is, I mean, in such a simple statement, what is for example, the spatial variation around the world. I should sort of also, I have this general slide to connect the fact that of course climate acts at multiple temporal scales. Where I refer to climate variability, it will be largely interannual variability causing multi-annual cycles in scales to 2 to 8 years. Characteristically, we also have seasonality, etc. So there are these indirect effects. And I like to mention today two sort of polarized debates. One that I thought was productive, and one that was, I don't know, something that should have been solved in 1 week and took 10 years. So it's interesting why. And so the first one is on climate variability versus herd immunity as a cause of multi-annual cycles. And the second is about climate change versus other trends, in particular development. And I should point out that it is interesting in the history of debates to find that they don't go away. They just come back under different faces. It's almost funny. If you read the early literature on malaria, there were these people that were really at each other, the contagionists versus the localists, that thought that environment versus transmission explained the patterns of malaria, of cholera. In fact, the books on this were really, really bad about, the comments about others. We had our own nice discussion in the 1950s that was big in ecology, discourse of importance of density-dependent versus density-independent factors. That is very much the same question that became then the question of chaos versus noise in nonlinear dynamics, that then in disease comes back, but later. In the 1990s, it's the same question, what was called intrinsic dynamics versus extrinsic climatic factors in epidemiology. So it's interesting, like for some of you who are younger and may not appreciate the importance of some of this debate, I just have on the density-dependent versus density-independent factor, of course, the work of these entomologists in Australia, who very strongly thought the environment was extremely important. In fact, they said the density-dependent factors do not describe any substantial body of empirical data. And that is a wonderful book, big like this, full of empiricism. Now, what do we mean in diseases when we think about intrinsic versus extrinsic factors? What we are interested in that case is to explain the multi-annual cycles that you see in seasonal epidemics. So the periods longer than 1 year, for example, here in cholera. And the question is, for example, climate variability driving these cycles? When you get to the literature, this is early 2000, this was an influential review, where you read these kinds of reactions to work on, for example, El Nino driving some of those cycles. The authors write, longer-term weather cycles, such as ENSO, have been invoked recently to explain outbreaks of malaria and other diseases. None of this analysis allows an alternative explanation involving intrinsic cycles. And this appears in a number of papers that claim, for example, for both dengue and also more theoretically in malaria, that you had this resonance of herd immunity with seasonality. So I get to what they mean by this. Many of you know it, but it's kind of interesting. We all know SIR kind of dynamics in childhood infections or COVID these days, like populations acquire immunity. You got this tendency of the population to oscillate because of this variation in immunity levels, and then the birth rates replenishing the susceptibles. If you add seasonal transmission, you get a forced pendulum, because now you have two time scales, the one of transmission, the seasonality, and the natural tendency of the system to oscillate. You can get anything you like as a function of parameters. A very elegant example of measles, and some of the studies of time series of cases pre-vaccination show this small peak, large peak, small peak, large peak. You can say, is there a two-period cycle anywhere in the environment? There isn't. It's a purely emergent cycle that comes out of this. So when they talk about intrinsic cycles, this is what they mean. It's not purely intrinsic because you have seasonality, but it means that it's an emergent pattern that has nothing sort of driving it exactly at that scale. Now, the problem with that is, how do you essentially test for this alternative hypothesis? And I will say that what has been useful about this is that out of this debate came a series of methods in population dynamics that you can use not just in epidemiology. You can use it for other long time, well, not so long, but for time series data, when you want to basically confront two hypotheses that you can write as models. Stochastic models that can have not just observational noise, but hidden variables. And there are a lot of very interesting statistical inference methods that we can use these days, including very nice packages to sort of do this kind of inference purely on the basis of stochastic simulations and methods to estimate likelihood, do model comparison, etc. So it was useful. You can use your hypothesis and see which model better explains the data, and even if just you have a noisy observed variable, you can test this. I like to show two very. I would tell you that I chose this, so there are worse results, but I get to choose, so I'm choosing you some interesting ones. So this is an example of rural malaria in a district of semi-arid India. It should relate trivially to rainfall. The data are in red. But what you can do, these are cases of Plasmodium vivax malaria. But just for the point I want to make, you can actually fit your model. The model that is fitted is simulated from estimated initial conditions in black, with the noise in brown here. This part is purely prediction. And what is interesting out of the prediction, what is interesting to me is, this is interesting to, for example, control people who are intervening. You could look at this and say, well, the epidemics went down. Is it because of climate variability, or is it because of intervention? And this is one approach that you can take to say, in this case, your effort is not really the answer. In this case, the climate conditions, the drought explains the pattern. So to consider intervention in the context of climate variability, these methods are useful. And this is another very chosen example. I just like it because it's an example of fitting the data, again in black, for two cities. This is urban malaria in India, in two cities of India, but just to show a model of transmission of malaria. This is falciparum malaria, but driven by humidity, for example. And I'm making the point that these methods are useful for prediction. This is completely out-of-fit prediction, 1 year at a time. And you can get very nicely this multi-annual variation. So we have now tools that I think those kinds of dichotomies of is it the nonlinear dynamics of the disease, is it a climate factor, you can write it as a hypothesis, you can use this nice machinery and try to get some answers that I think are useful. So that was useful. In the case of climate change, it's a little bit more complicated. I should say partly if you work with a trend, it's always a mess because everything has trends and it's very, very hard to really attribute the change. So, and there are many other important aspects of change that are important and that you have to consider. So this has been a problem, and I'm going to talk about a debate that happened about the importance of climate change to highland malaria, to malaria in regions at altitude where common sense should tell us that climate should matter. First of all, if you ask yourself, where should you find the clearest evidence of climate on malaria, or for that matter, on other infectious diseases, in the places where climate to begin with is a very strong limiting factor. So this is at the edge of the distribution of the disease, either in altitude, in latitude, at the edge of deserts with water, etc. And this in red are the regions in Africa that are at the edge of the distribution of endemic malaria, that are the areas known as epidemic malaria, where we expect a signal of climate change. And an example of the cases reported from the 1980s to here the 2000, this is for two parasites of malaria, where you see this increasing epidemics as you go into the 80s and the 90s. Very controversial patterns in terms of the role of climate change. And in fact, that was in Ethiopia, where there are two parasites. This is in Kenya, where there is only falciparum, but 2 time series in some tea plantations that go to the 1970s, and that became the center of a lot of the work. And you can see here again the epidemics in the 80s and the 90s really taking off. So I apologize for the next slide. You don't need to read it all. This is my brief summary of the timeline of why did it take, 10 years, right? So what happened to this debate? So the question was whether these increased epidemics were caused by climate change, by warmer temperatures. There was a very influential paper in 2002 by Hay and colleagues, and many other papers that then relied on this finding, that there was no evidence of statistically significant trend in a time series from this gridded product. This is from the Climate Research Unit time series, which are a gridded product at half degree latitude longitude. But anyhow, they looked at the data up to 1995, and they said there is no statistical significant trend in temperature. So why are we trying to explain an effect when there is not a trend? At indeed an NCIS meeting, we looked at this data with updating it to 2002, and looked at this version of the data set that had been at the time what you could download it. And for those of you who now can find gridded products very easily online, at that time to download those points in the grid, you had to go to some unofficial Fortran code that was provided at your own risk. And whatever, it took a while, and you had to be sure you had the right box. Anyhow, there wasn't an easy way to get the data, and on top of that, I was downloading this version. So we basically did a different analysis, which we thought was more robust, and we basically concluded that there was a significant trend. We were unable to identify at the time the reason for the discrepancy. This was not nice, but we thought we should publish it. There was more to the paper. There was an effect with a mosquito model, and Jonathan wrote an editorial, so that was a nice thing. And then, so we offer some discussion on the methods, on why we thought our methods were better. There was not, after that, a lot of progress on what was going on, and it took several, really up to the end of the decade, to basically find that there was this clear evidence of warming trends in the updated products of this gridded data set, and that, of course, there are similar warming trends in many other highlands. And in fact, if you looked at the reason we did the analysis was because when we downloaded the data for something else, we thought if there is not a significant trend here, there must be a problem with statistics. Now, it was interesting that these are the 4 sites, and so you can say, well, what really happened? In fact, the same authors, with an additional here first author, wrote a paper in PLOS ONE in 2011, pointing out that there seemed to be a difference in the data sets, in the gridded data sets. And they had a long paper about this and other things. By then, other people had pointed this out. And I wrote, it's nice in the PLOS journals, you can write a commentary back. I thought I should respond because this was presented as, there were these different data. And there was a lot more to it. So I wrote that it was unfortunate that almost a decade was lost with no clear resolution on an important empirical pattern that could have been clearly recognized by the end of the 1990s, beginning of 2000. And so perhaps some lessons can be learned for other work. And one would hope in particular that open discussion of scientific results can prevail in areas whose relevance goes well beyond science and academia. Now, what was interesting is that if we use, at that time, we had access to the same data sets. If we use the same method that was used originally, but with either 3 more years or 5, we changed the window to which we apply that method, we would have found significant trends, just with a few more years. So it wasn't like because we went up to 2005. It was really the window of time that was analyzed. It wasn't even the method. So it shouldn't have taken that time to tell this. And let me go on. Of course, you can say, well, it's obvious that we need better and easier access to climate and disease data, especially, of course, good data. Climate, I think that is easier. I think the disease data, contrary to the fisheries, is much less available when there are a nice retrospective time series. They are invaluable and they are countable. Now they are becoming more available for some diseases. But they are incredibly important. What I think there wasn't at that time was a better setting to identify that, okay, there is this conflict in these results. Can we find a setting for working together on what was going on with this issue? Because after all, we are talking about climate change and malaria, which was the big poster child for the effect of climate change. So even if you don't care about the highlands. Of course, we learned a lot about the difficulty of addressing trends in time series that have multiple temporal scales of variation. And this was why their method had failed. And if you place the window 3 years over here or there, you would conclude something different. So a little bit of interannual variability can really mess up some of these things. But really, the question here in red, maybe you cannot read so well, is not really whether there is a statistical trend in temperature, which you now are sitting there thinking, how could that be contested? It was at that time. And you can say, well, the central question is whether the variation in the temperature can have a biological effect. And interestingly, the solution to that, there were other studies, including mine, but I thought that the one that gave the most clear answer was a very nice data set, because it was in two regions, two data sets, two regions in highlands, one in Ethiopia, one in Colombia. And what we had for these periods were monthly data in space and time, but at very fine spatial resolution. And so therefore we could look at what happened with climate variability. And I'm not going to give you the analysis. This is just to show you this place in the highlands of Ethiopia. The point is that when there was an El Nino year, you could see this expansion, both where the disease was already, and then it would expand to higher altitudes. What you can do is then compute this altitudinal expansion, how much of an increase it represents in space, and then see if it was comparable to the what you expected with the trend. So if these things were consistent, you could go from climate variability to the trend. And in reality, you can say these were small regions, but if you look for the whole of Ethiopia, where there is a lot of population at these higher altitudes, we basically extrapolated that an addition of 5 to 6 million cases were added from the 70s to the mid-2000s because of the climate change that had already occurred. So if these kinds of expansions with altitude were occurring. And what I have here in my commentary was a comment to say that now that we know that climate was having an effect on this up to the 90s, people were losing interest in this question because we were observing that the epidemics were now becoming smaller at the beginning of 2000. And therefore, I was saying, well, it's too bad that because of the delay in reaching a consensus, now the issue of warming trends doesn't seem to matter because we are intervening, and if we are intervening, the epidemics are going away. All those places, because they are at high altitude, have epidemic but low transmission, so they are places where intervention can be fairly successful, by contrast to what I showed you yesterday. So I'd like to show you something that I thought was quite interesting that we did recently. I know I shouldn't show you research, but there will be no analysis. The point is that, yes, in the years 2000, these are the same region, both parasites, the same time series, showing that the epidemics, start turning around. So we asked, could this be temperature? There was at the beginning, at the turn of the century, there was something that was called the slowdown of climate change. This is by no means a slowdown of climate change. This is a transient, decadal pattern in climate, which was very interesting. I think it's not in our radars because we think a lot about the scales of El Nino. These are decadal time scales, and there is a lot going on with decadal decadal variation in the oceans that is deeply connected with what is happening with climate change. And it's a mess. It's changing it's related to El Niños. It's changing how different oceans are interacting with each other in the weather. But what was interesting to us was a very simple question. People said, well, this is intervention. And we said, could it be that this disease is so tuned to climate that it's responding to the slowdown of climate change? And this is just the trend, the same trend we took from T-min there, T-minimum temperature. You can do it with the mean, just to illustrate that at least there is a correspondence in the trend. That's not good enough. So with the same tools we I told you were these nice tools, we could do this counterfactual experiment. I give you the red data, you fit your best mole up to 2000, and you say if nothing changes, but we look at the effect of temperature, what would you predict? And what is interesting is the predictions are in blue, the median prediction, because this you have to simulate stochastically. And in fact, we are simulating from time zero, and then that's the median, and then there is this uncertainty. And the red are the cases. You can see that at the beginning of the century here, we predict quite well. I mean, this is messy, but we are predicting quite well. When things fall apart, it's exactly the time when very strong interventions were brought to this region. So the reality is that this decadal change in temperature was helping intervention. So intervening here may have been harder. And so that is kind of interesting, and I'm going to skip this. If you're interested in the paper, Javier Rodó, who is my collaborator and a climate person, did a bunch of stuff. I'm not supposed to show you research to show that in fact, the connection of the regional temperatures to the El Nino was changing across those decades. So there is these global changes that are happening at these multiple temporal scales. And I'm not going to read all these, but I just want to say the coupling to climate is in fact so clear in these regions that you see that seasonal, interannual, and these decadal timescales. This introduces this big problem, I suppose, also in fisheries, where you had a lot of these multi-annual, there was a signal of interannual variation, because we have to intervene in the context of these longer cycles. And we have seen these examples where you think you are doing great, you are intervening, so the whole system of public control relaxes because you think you no longer have a problem and you don't have the resources to keep it up. And so there is this very interesting feedback. And so you are doing that in the context of this crazy variation at these, multi-annual timescales. I think that kind of picture is, so if we return to this idea that basically says, global warming does not matter because, things are going to get better with intervention, I think we are missing an opportunity to understand how to inform intervention with what is going on with climate. But because this is about maps, the whole thing about time series sort of went away, and then there were questions about this global spatial distribution of the disease, which really this article was about. So let me just take 5 minutes, and talk about the maps, because basically this was an interesting study looking at the map of malaria, an approximate map of malaria, done very early on at the beginning of I forget the exact date, maybe early 1900s of some sort, but I'm just saying it was compared to the map they got in 2010, and they produced maps with some interesting approaches, not just for malaria, but for many other things. And here is a comparison of the early map with presumably the current map. What these maps are, they are using all the prevalence you can find around in the literature, in all the data sets, and they are doing some elegant, I would say at least technically elegant, interpolation with some Bayesian methods. But the conclusion, of course, you can look at this and say, well, the regions in green in particular of high malaria are getting better. So this is what is behind that editorial. And if you read the text, you got these conclusions that say there is a substantial weakening of the global correlations between malaria endemicity and climate. So the idea in the paper was there is no connection anymore to temperature because intervention and development is blurring the picture. We tried to respond, to ask, and this is the only thing I want to show about this problem of we tried to respond what was happening in the regions where we knew climate mattered, not around the whole world. In fact, the map looked a lot better because malaria was controlled at high latitudes in the temperate regions, not in the temperature regions, at the edge of the latitudinal distribution, where the vectors are not as good as the African vectors. They never were. So if you compute indices in that map that have to do with area, you do very well. But if you are interested in particular areas and you are doing public health in the highlands of East Africa, then you may want to know whether this conclusion applies. So we look at the surveys that were used. This is from Kenya, for example, and we decided to look at one thing. There were places with a low density of surveys, here in yellow, and places with a high density of surveys. Because we were concerned that if you have elevation, that is your measure of temperature, and you have sparse surveys, you are going to interpolate something that has no signal. And so we basically looked at this in Tanzania and also Ethiopia, and I will only show this graph. This is altitude, this is prevalence. The black dots were just for check from an elegant study by Cox and colleagues, where they show this very characteristic pattern of decreased prevalence with altitude. The red is from the box with high surveys, and the yellow is from the nearby box of less surveys. My point is, yes, there is no effect here of temperature because you are not sampling enough. Otherwise, the signature is there. Now you can say, okay, these regions are small. I will not, okay, this I already said. You either need to include denser surveys or include temperature in your model for these regions. But what is interesting is, of course, more generally, you can say that if you are interested in public health intervention, you may be interested, if you are sent in the World Health Organization, maybe on the global burden of malaria and climate change. But you are also, I think the intervention has to be addressed in regional scales. And at regional scales, we cannot say that the effect of temperature is gone. And these highland regions have some millions of people. They are not a few people. So I'm just saying that's just a perspective. And let me end with this. I like this movie, so it's a bit, let's see if I can play it. This question of development, right? Development in many places around the world is closely connected initially with land use change, either in places that bring irrigation to arid regions, places that deforest, etc. And I have this just because it's, you know it, but it's shocking to see. This is the state of Rondônia in the Brazilian Amazon, and you can see the deforestation that goes on to bring this in a few decades, right? The loss of the forest. And so you say, well, development is going to make things better for malaria. As we know from work, including by Jonathan here, on what happens when you start doing this, initially the risk of malaria increases. And there is a substantial body of work that shows that eventually development may get you to a better place. Eventually, it's not necessarily the same people, but you have to go through this transition that can take of the order of a decade. And you may also get stuck in conditions where things do not improve. So we wrote about this with a model, about this pattern of development in a model that had an economic model coupled to a malaria model and land use change, where you basically show this behavior that people have described empirically, where you get this rise and fall with so-called development in these areas. So the question is, do you want to go through this trajectory and take a decade of worse conditions for some of the population? So I'm just saying, I'm posing this because now I like to sort of conclude why is it that some debates, after all, we do this all the time, dichotomies are great to organize our thoughts and to basically formulate hypotheses and in opposition. The problem is when we take them too seriously. They are not necessarily meant to be that serious. They are often a context for finding the nuances and the interaction. We were, in a sense, I think in this field, I tried to give an example of where we failed to see it like that and to discuss the nuances of the kinds of editorials and the kinds of conclusions that were taken from the results. So this is either in the media or even in the scientific editorials. And it's interesting to me that the area where this happened, it was not random. It's exactly where you know that the science is most influenced by non-scientific aspects. And so it was a very interesting experience to see the degree, because of course, and I benefited from that, so I'm not going to complain. When you have that kind of pendulum, like it's this, no, it's this, and it's yes or no, which of course it's too simplistic, the papers get a lot of visibility, you get a lot of attention by the press, you never know what you should say because it becomes a simple story. But so that kind of attention really, really generates a situation in which it takes a long time to make progress on something that should have been self-evident. So I guess I don't have a lot of, I was supposed to talk about something about practicing science. This is no longer a debate, but many things are. And I think like we run this risk of playing into the hands. I don't need to read these things. There is this great, there are all these disastrous websites. You can read some of those if you like. We also may get even, feed material to that kind of word. So there is a responsibility of how we go about these debates. And you can say, well, how come you couldn't tell you had different data sets? Well, the conditions at the time where it was not so open to share the data made it particularly difficult in that example. But also this sort of climate where it was so important what you found made it also difficult to have the kind of dialogue we should have. So I'm not sure that helps a lot. I thought I'll share that with you. I like to thank these graduate students and then my long-term collaborators on both the statistical methods over there on the left and then some of the climate and the epidemiology. Thank you very much.
Questions?
Yes, Jonathan.
So, Mercedes, I have to ask this question because I remember being in those trenches back then.
You were in.
So scientists, every scientist has bias.
Yes.
And bias is not a bad thing. We don't do science unless we have some predilection,
yes,
some beliefs that there's something that we want to show.
Mm-hmm.
And so scientists come in with bias. And on the one hand, we're living in this day of disinformation and just nastiness. But in your story and in this debate, we have good scientists, very good scientists on either side, bringing their own predictable biases. And I'm wondering what you think may be a way to inoculate against that type of repeated situation where, scientists come in different, but to really be objective and to see the literature growing and changing. What was that particular situation where you think there could have been something to fast track that argument and resolve it more quickly?
Yeah, no, that's an interesting question. I don't know a lot. I think to answer this. The predictable biases, of course, we do have them, but in the end, we are still doing analysis that we hope are as objective as possible, right? I don't think anybody was hiding anything. I thought there was a lot of misunderstanding, but so I think the biases are one thing. I think what was happening was that there was a historical, almost like a response, right, which was historical. It was like, okay, there has been all this literature saying climate change is terrible for malaria. So suddenly, if you found evidence that this was impossible, there was a very high payoff in terms of the attention to the research. And the same if you suddenly then came and said, no, that cannot be because now I'm finding this. So I'm just saying that we have to be more careful than with our biases, like the kind of this kind of pendulum and the kind of payoff perception that could lead, I think, good scientists to see a result and then it's, it gains more attention perhaps than it should. And of course, this wouldn't happen so much today because it would be easier to hopefully with more open data sharing, this would be a lot faster, right? In terms of finding resolutions and finding where the problem was. The biases, I think you have to be careful that you don't. I don't know if you can say it's predictable what the biases should be, right? I think we didn't go into this necessarily. I don't think neither of the groups went with their predictable bias. I think you went with a history of something that you thought must be wrong and you were going to check. And so I'm just saying that this that in particular, and the reason I showed the editorial was just one thing, but I'm just saying there was a lot of press for these results, and I think that reinforcement is what we have to be a bit careful because I think it generates a situation where we take these dichotomies too seriously rather than looking at finding some sort of consensus or even in what ways you cannot simplify these to it's one or the other.
If I can just say one thing, I think that the more interdisciplinarity you have, the more experts in the room on an issue, the better. And back then, because climate change was such a polarizing issue, that I think that without having climatologists working with the malariologists, I think that was part of the problem.
Yeah, that was a very good point. I had it in a bullet point that I didn't really read, which I thought was interesting because I completely agree that if you had more climate people at the table, maybe they would have said right away, this is where the problem is. Certainly now there is more collaboration of that kind. And interestingly now there is a discussion about effects, but there is not a discussion on whether there is enough change that we may see effects. And so I think it's interesting how certain debates parallel the state of things, outside the scientific community. Like it wasn't by then very much accepted as it is. There was a lot more doubt about, at that time, going into that time on effects of climate change and even the extent of climate change. So yes, I agree that the more, in particular also working, of course, with the more quantitative people, the climate people, you definitely need those groups. And a solution is, of course, for some of the things to create the venues for the very direct interaction of a group of people with the data, which I think for some reason never happened in a reasonable time, which was part of the problem. So.
Question.
Yes.
thinking about the time scale on which this debate unfolded, and then thinking about the pandemic we've been dealing with recently, where you have this sort of bewildering amount of data at everyone's fingertips, seeing case numbers and strain types coming at a daily scale. And you don't have the same types of problems there, but I think when you think about that window of your perspective of your data set and how you're shifting that window over time, what do you think? Do you think there are different issues at play, or do you think that this sort of open science, everything's freely available at such a fast pace, is the solution to that? Or what's your take on that?
Yeah, that's a very interesting question, because I think that for COVID that posed a different problem, because we had too many people and too much science being published too fast. And in fact, some of the reviews were so fast that, I'm not sure under normal circumstances the same papers may have made it, in the state they made it. But so these things were coming, and I think what would be very interesting was to do some sort of phylogeny of the parameters people started to use. Because there were some, obviously people were calibrating and fitting, but there was like, okay, we know this parameter, we cite those papers. At some point, the evidence for the kinds of modes or parameters, it was hard to know exactly where things came from. It was an overwhelming amount of literature. And then, people were publishing upon published parameters by someone else, and therefore it was channeling, the modeling in particular ways, which was a very interesting problem. It's like it was almost impossible for a human to kind of follow what was happening, which group did what, which parameters came from where, and that I think was a big problem with this. There is this question of like, did we need all that effort on that amount of data, right? There were some very good groups I think we needed, but I think we had so many models and so many fitting exercises that it was, very, very hard to know. So there are, of course, these ensemble models and ensemble predictions which attempt to do this, but again, you don't need, as many. The effort was overwhelming. So I'm not sure what the solution to that is, but in response to, it's a completely different problem. Still, for the very long time series, I think we still lack in epidemiology a consensus on how we are going to share data, because partly the partners that are providing the data, of course, are collecting it for public health purposes. There are people in other countries, etc. So the issue of publishing and sharing your data, at least for some of these long time series, is not as simple as we would like it to be. And there is not, I think maybe in climate there is an agreed way of sharing data and window of time for sharing data. For epidemiology, we have some rules by NIH if they fund you, but it's a question whether people really can always follow this because of the restrictions of their partners. So we are, I think for COVID there were a lot of data, and that was good. For most other problems, we don't have enough data. I mean, we have these possibly nonlinear systems on top of these scales of variability of the environment, and we are dealing with countable data sets compared to what people do in climate science. And that is a big problem. And I always say, noisy data is a lot better than no data. So when people say there are no good data, I think it doesn't matter. If we could mine a lot of the data that has been collected for other purposes and share, we would make a lot of progress. And that is still something that we are not there. Yes, Tony.
So when you were talking about the history of this, you brought up Andrew Arthur and Birch.
Yes.
And density dependent, density independent. That was a discussion that was really about the basic science. When you talk about malaria, you're talking about a disease, policies that affect hundreds of millions of people. Do you think the issue of consensus and disagreement in science needs to be different when you're considering very basic science results versus those that have impacts on human health or societal matters that really make a difference?
Well, let me think about that. It's a very very interesting question, and I definitely think that influences not the how the debate on those issues, at least by some of the parties, was incredibly strong, right? But of course, the field moved on with very interesting ways to look at the interaction, to look at ways to test these things, and there were a lot of methods that came out of it. So that was great. I must say, of course, when you think of public health, even fisheries, right? You are in a domain where there are other parties interested in the answer, but I don't know that we have to be any different about the science, right? And about how we try to become less biased, right? Because I think we fool ourselves if we think that my little model is going to change a big thing in public health. That's also true. What it may do is, go to some IPCC report as a piece of evidence. It's just one more brick in the whatever. And then, yes, if you are going to offer your predictions for a particular purpose, you want to sort of use as best as you can, these issues, look at uncertainty and so on. In terms of climate change and malaria, there was this problem of like the first maps and the reaction that came was against maps of suitability for the vectors, right? And if you read those papers carefully, people were saying we are considering the future only as a function of temperature. So it was a little bit unfair to then say, well, but they have not considered all these socioeconomic things. The problem is, I think the difficult part and the responsibility is when that gets outside of the science and portrayed outside of the science as we are going to be in a world ridden with malaria, right? Or things like this. But clearly, we have effects of climate change on other vector-borne diseases that are emergent diseases like as dengue and so on, where in fact the spread of the vectors and the suitability are a problem. So there is this interesting question about how your findings are going to be interpreted more than really used for, a lot of public health. And I think that's where we have to be more careful. It's sometimes very difficult because you are neither in control of the editorials nor in control of what the press publishes. And that is a very crazy experience, like where it's partly a learning process, but it's really not in your control. So it is unfortunate, but that's a little bit where perhaps, we have to be extremely careful because it's different to talk about insects in a debate about, the Nicholson blowflies and but again, I try to point out, and it's interesting that climate variability, after all, led to a lot of improvements. It was, I think, climate change that was where progress was stuck, and exactly, I think it's more the difficulty that those topics pose, even in the science, than really this issue of responsibility, because I would hope that the science is the same in the end. Sorry, I went in circles because I'm trying to think about what I'm saying and I'm not very clear. Okay.
Okay. Well, thank you so much. That was wonderful. And before we move over to refreshments and snacks, I just want to thank you all for coming, both online and in person. I want to thank the speakers on both our days, did a really wonderful job. And thanks to my host, sort of, and Kyle, who did really the lion's share of the work putting this together. So thanks so much for that. Yeah.
What did you do?
No, I was kidding.
It's a bunch of fun. I didn't see it soon.
I realized what I was going to be doing. I thought for